So it's my absolute great pleasure to introduce Viola. So Viola is a PhD student with myself and with Jan Buitelaar, and she's based at the DOMS Institute. Many of you will know Viola as she's heavily involved with testing and analyzing data for both LEAP and PIP studies. And her PhD is focusing really on multimodal measures of excitation and inhibitory mechanisms. And so today, Viola is going to be talking about that work, looking at excitation inhibition and looking at the links between brain and behavior by combining different methods, including genetics, MR imaging, as well as behavior measures, focusing on the LEAP cohort. So Viola, take it away, and I'll keep an eye on chat.
Thank you so much. I'm going to share my screen. All right. I'm just going to minimize you. All right. Well, thank you so much, Nick, for the introduction, and thank you for the opportunity to let me share my latest work with you, everyone. So I want to start here. We know that our experiences of the world, our social interactions, and our behaviors comes from our brains and how they function. And the key cells in the brain that makes this happen are neurons that connect and communicate with each other. And there are two ways that they communicate. They're either excitatory or inhibitory. And this is done with neurotransmitters that have one of these two properties. The balance and the interplay between excitation and inhibition determines brain activity and eventually regional output. So the balance between excitation and inhibition is important and affects functioning of the whole brain. And changes in this balance, so an imbalance, leads to differences in how the brain functions. So examples where this happens is epilepsy or Parkinson's, where we know how an imbalance might look in both of these, and we also know where in the brain this is probably happening. When it comes to autism, an excitation inhibition imbalance is a popular and widely accepted theory of what in the brain is underlying it that leads to the differences in behavior that we see. And this makes sense when, for example, you know that epilepsy is a lot more common in autism, but there's also a lot of other evidence, mainly coming from animal studies, but also in studies with humans. But we don't really know yet what kind of imbalance this would be. Is it more or less excitation or inhibition? We also don't really know where in the brain this would be happening. But autism is very heterogeneous, right? It could be that there are different kinds of imbalances in different parts of the brain that links to different behaviors that we see in autism. And if we can find these links and understand what is happening in the brain, that could be helpful for developing more targeted support options. So for example, if you are an autistic person struggling with certain sensory inputs or environments, knowing what is happening in the brain is useful for finding out what support options are most likely going to be helpful for you. So we wanted to find more direct links between excitation and inhibition in the brain, how it relates to brain structure and to behaviors in autism. And we did this here by combining genetics as a proxy of excitation and inhibition, brain structure measured from MRI, and behavioral measures. So because I will talk about genetics, I want to start with a brief introduction to genetics, just so that everyone is on the same page. Every part of our body is made out of proteins. They are the building blocks of life, essentially, and the proteins themselves are made of strings of amino acids. And sequences of these are determined by our DNA, which is sort of the code with the information of what proteins to make and how. Most of the DNA that we humans share is identical. 99.6% of the genes in any person's genome is exactly the same as in any other person. But there are, of course, differences between us. And the 0.4% that is left is what contains the genetic variation between individuals. And within this 0.4%, every person has about 4 to 5 million different variations. And examples of people having different genetic variations that we can see very visually are, for example, different eye color or hair color, or if you have straight or curly hair. If we zoom in on a DNA strand like we've done here, we can see these parts with the letters that you might have seen before. These letters are paired since our DNA consists of 2 strands, and these pairs are referred to as single nucleotide polymorphisms, or SNPs. And the combinations of these SNPs together encode for all the amino acids that make up a whole protein. And that is what a gene is. It's a bit of DNA that encodes for a certain protein. And differences in SNPs is the main place where genetic variation occurs, so the majority of that 0.4%. And this is also then how genes encode for certain functions. They encode proteins involved in specific processes and functions. So if you want to relate this specifically to the brain, all the cells and their parts, like the receptors and transporters and the neurotransmitters, are made out of proteins. And different SNPs, so genetic variation, can then lead to different functional consequences. And this also means that genes encode for proteins involved in excitatory and inhibitory neurotransmission. So understanding genes can help understand mechanisms of how the brain develops and functions. Every cell in your body contains all of your DNA or your genome, but different genes are expressed in different cells depending on their function. Specific parts of the DNA, so the relevant genes, are expressed, which encodes for the proteins needed for a certain function that the cell will have. So what's originally one cell in the uterus gets copied, different genes are turned on in different copies of that original cell, and you get the different cells that make up your whole body and your brain. To know what genes are expressed in an individual cell, there are a few methods to go about it. And one method that I want to focus on here is using microarray techniques, which is a fancy method that I'm not going to go into detail about. But we can use it to see what genes are turned on in what cell, telling us about their functions. To do this, you need to take the brain of an individual and look at very thin slices of tissue, extract the information from the cells, and analyze that information to read what genes are expressed or activated in the cell. That means looking at the individual cells across the brain to see what genes are expressed where. And as I think you can imagine, this is a lot of work. And you can only do this on post-mortem tissue or in animals or in Petri dishes, since you need to slice the tissue. Thankfully for us, this has already been done in humans, and the Allen Human Brain Atlas has this data freely available from 6 donors, which we're going to use later. This was then a brief walkthrough of genes, what they are, what they do, and why investigating genes is relevant for understanding the brain. Now, if you want now we know that genetic variation are variations in the SNPs that are parts of your genes, and that gene expression is the process of genes that are expressed or turned on to encode proteins in any individual cell. We also know that genes encode proteins involved in brain functions, since they're part of the development of every protein. So to start using genes to understand excitation and inhibition, we can then select genes that we know encode proteins involved in certain functions that we are interested in. Glutamate and GABA are the most common excitatory and inhibitory neurotransmitters in the brain respectively. And typically when we're doing in vivo measurements of excitation and inhibition, these are the ones that we look at. So we can select genes that are involved in the glutamate and GABA pathways in the brain. We selected our genes using Ingenuity pathway analysis. This is a program that collects information about genes in a canonical way, meaning that they compile information about the functions of genes from literature. And this includes gene expression data, annotation databases, studies looking at up and down regulation and things like that. So they gather the information about which genes are more than chance involved in certain functions. A pathway of genes is a whole pathway of a biological function. So take, for example, glutamate pathway signaling in the brain. Ingenuity pathway analysis selects all the protein encoding genes for this glutamate pathway. This is, for example, genes involved in the pre- and the postsynaptic neurons, which you can see the outline of here. And glutamate itself, it's not a protein, right? It's a neurotransmitter. So genes don't encode for glutamate in itself, but we select genes that's involved in the glutamate pathway. We ended up with 72 genes in our glutamate set and 124 in our GABA gene set. And the work that I'm going to talk about today was published earlier this year. And in it, we list all the genes that are included in these gene sets. So if you're interested in looking into this in more detail, I will give a QR code to the paper at the end as well. So now we have our sets of glutamate and GABA genes, and we will use these gene sets to investigate genetic variation of glutamate and GABA to behavioral measures. This analysis method is called competitive gene set analysis. And we will also look at the expression of these genes, combining gene expression data from the Allen Human Brain Atlas with cortical thickness measures in our own participants. And this method I refer to as gene expression analysis. And I'm going to explain these methods in more detail soon, but I want to already now mention here that these analyses that I have used here are in no way possible to use as some kind of genetic diagnostic marker or anything like that. We cannot use this to single out individual genes or individual people based on their genetics. These are purely methods to investigate glutamate and GABA functions in the brain by looking at how genes that we know play roles in the glutamate and GABA functioning relates to other brain and behavior measures that I'm going to talk about. So before I get into the slightly more technical parts of the analysis methods, I want to tell you about the data that we used. So first with the genes, we selected genes that we know encode for excitatory and inhibitory mechanisms in the brain, and that can tell us about relationships with other measures like structure of the brain, where we have good evidence for there being differences in autistic people. And because we ultimately want to understand how this relates to the behaviors that defines autism, we also wanted to associate it to behavioral characteristics. The data that we used came from the Longitudinal European Autism Project, or LEAP, and here we used data from wave 1, where we had data from 638 participants from Nijmegen, Utrecht, Mannheim, London, and Rome. And participants were either autistic or non-autistic and between 6 and 30 years old, which will become a bit more relevant later. Starting with the genes, like I mentioned earlier, the most common excitatory and inhibitory neurotransmitters in the brain are glutamate and GABA, and we selected genes that we know encode for proteins involved in glutamate and GABA pathways in the brain. And these gene sets that we used have been used in several studies before, and we use them as our proxy for excitation and inhibition. Then we wanted to look at brain structure, specifically cortical thickness, which essentially is a measure of gray matter volume across the cortex, which is the dark gray parts here. Because in the brain, it is in gray matter that neurons connect and communicate. So this is where most of the communication between neurons occur. And the differences in cortical thickness has consistently been found in autistic people, where both increased and decreased cortical thickness have been found in different brain regions. And this could be affected by glutamate and GABA functioning, since they play a role in the functions that goes on there, and both have been linked to autism previously. We can measure cortical thickness using MRI, where we make an anatomical scan and then we extract the cortical thickness. And here we did this using FreeSurfer, where the images were modeled and parcellated into 34 brain regions per hemisphere. So we have cortical thickness for each brain region in every participant. And then to capture the behavioral characteristics of autism, we included questionnaires that tried to capture the core features from all of the participants, and we also had diagnostic interviews as well. So with the questionnaires, we tried to capture repetitive behaviors from the repetitive behavior scale, social behaviors from the social responsiveness scale, and sensory processing from the short sensory profile. And then the diagnostic interviews, which were only done with the autistic participants, were ADOS and ADI. So together we then have genetic data where we focus on glutamate and GABA genes, we have cortical thickness measure, and we have the behavioral measurements of autism characteristics. For the analysis, we used two methods that I'm going to get into now: competitive gene set analysis, which looks at relationships between genetic variation of glutamate and GABA genes and the behavioral measures, and the gene expression analysis, which investigates correlations between the expression of these genes with cortical thickness differences between the autistic and non-autistic participants. So now there will be a more technical part for those who are interested, but I will also summarize what each step is actually doing in a less technical way as well. But in general, it's not necessary to understand every detail of these analysis steps to understand the findings that I will discuss as well. Starting with the competitive gene set analysis, we did this using Magma. This is a gene set analysis tool that we use to associate genetic variation of our functional gene sets, so the ones that we selected for glutamate and GABA earlier, with a phenotype of interest, and here that is our behavioral measures. What makes this competitive is that it compares the association of the aggregated genetic variation in our whole gene set to the association of the rest of the genome with our phenotype of interest. Several kinds of tools exist to do gene set analysis, and they have slightly different ways of doing it, which essentially can lead to testing different hypotheses or having different sensitivities or assumption. Magma has been shown to be more robust against biases like gene size, since a larger gene means more SNPs, which can bias the results, but also to a number of participants, number of genes in the gene set, and it also accounts for something called linkage disequilibrium. Linkage disequilibrium means that the SNPs, or the genes that are physically closer to each other, are more likely to be correlated, as in they encode for proteins involved in the same functions. Basically, this means that not all combinations of genes are equally likely to occur, and this can create a bias when you're doing gene analysis, but here we take this into account. Competitive gene set analysis is done in 2 steps. First, we do a gene level analysis associating every gene to the phenotype of interest, and we get correlations between the genes, which we use to account for linkage disequilibrium. And in the second step, we take this output data, so the p-values of associations and the correlation matrix, to associate the whole gene set with the phenotype of interest. So this is essentially the equivalent of a t-test, testing if the average association of genes in the gene set is different from the average association of the genes outside the gene set. When we test the whole gene set that has the functional role, so here glutamate or GABA communication pathways, it makes the interpretation of our results easier than when we're investigating individual genes. It also lets us deal with genetic heterogeneity in the sense that you have a polygenic trait, so a trait that doesn't have a single gene that we know underlies the traits. So for example, with sensory processing, there isn't one gene for sensory processing, or more broadly, it could also be autism. So you have multiple genes that are slightly associated with the phenotype of interest, and these genes alone might not have a strong enough association to get over the p-value threshold, but when you group them together, it does, because it's the common function that is the causal effect. So here that would be the glutamate or the GABA signaling pathway having a causal effect on sensory processing, for example. The null hypothesis for the competitive gene set analysis is that the genes in the gene set are not more strongly associated with the phenotype than the genes not in the gene set. So this step involves a linear regression model on the gene level data matrix. The residuals are modeled as multivariate normal with correlation set to the gene-gene correlations computed during the previous step. This is what accounts for the linkage disequilibrium between the genes that are in close proximity to each other. And the gene set p-value is the one resulting from a test on the coefficient, testing the null hypothesis against the one-sided alternative. So in other words, what we do is test if the mean association of the genes in the gene set with the phenotype is greater than that of the mean of the genes not in the gene set. So these are the results that we got. What we see here is the list of the associations of all the behavioral measures with the glutamate and GABA gene sets. The ones marked in bold are the ones that were significant, and the p-values are the ones that come out of the gene set level test. And PFDR is then the p-value that we have after correcting for the multiple tests that we did with each gene set. So if we start by looking at the GABA association with sensory processing again, we see that after FDR correction, the p-value is no longer above threshold, but associations between GABA and sensory processing have been found in previous studies before, so it still seems quite reliable. And we also see that the ADOS and the ADI measures, so the diagnostic interviews that are mainly capturing other characteristic behaviors in autism, like repetitive and social behaviors, have associations with the glutamate gene set, but not with the GABA gene set. So together, this shows that glutamate and GABA genetic variations link to different behaviors in autism. And I want to note here that increased variation does not mean the same thing as increased concentrations of metabolites in the brain, but it does mean that variants of glutamate and GABA genes have a significant effect on behaviors. I also want to note here that these are quite a lot of tests, since one row in each table is one association. But most genetic studies have thousands of people in their cohorts. Here we have a bit over 600, which is a lot for the kind of cohort that it is, but for genetic analysis, it's not really. But as I mentioned earlier, this analysis method is less sensitive to sample size in terms of number of participants, so it's not really a concern for power of our results. Now let's get into the gene expression analysis, where we look at correlations between the expression of the glutamate and GABA genes with cortical thickness differences. And as a little refresh, gene expression is the process where certain genes in a cell are turned on to encode proteins for certain functions. This method was initially developed to investigate relationships between cell type gene sets and cortical thickness, and then they called it virtual histology. Histology meaning studying the structure of tissue, which makes sense when you're looking at cell type gene sets. We adapted this method and instead used our glutamate and GABA gene sets, so therefore I refer to it as gene expression analysis instead. And as I mentioned earlier, gene expression data is available from 6 post-mortem donors in the Allen Human Brain Atlas, and this is very extensive data, and the complete data set consists of more than 50,000 measurements of gene expression just from those 6 donors. So what Leon French and Thomas Paus did was use the fact that the gene expression data is presented as voxels with coordinates in the MNI 152 atlas, and they took this information and mapped it onto the DK atlas that is built into FreeSurfer. And they mapped the gene expression data onto the 34 regions per hemisphere, averaging the voxels for each region. So that gave us one value of gene expression per brain region per donor. And they also calculated the median gene expression profile across the donors. And this they have nicely shared for others to use, and this is also what I have been working with. If you want to read more about the Allen Human Brain Atlas to FreeSurfer process, the French and Paus 2015 paper is great, and there you can also find this data available. But to sum this up, we have gene expression data for every gene in every brain region, and it is in the same atlas space as the cortical thickness data that we have measured in our LEAP participants as well. So what we do here is test if there is an association between gene expression across brain regions of our glutamate and GABA genes with cortical thickness across brain regions. What I did more specifically was look at cortical thickness differences between the autistic and non-autistic participants. And to do this, we calculate cortical thickness difference profiles in each brain region and gene expression profiles in each brain region, and then we correlate the average cortical thickness difference with the average gene expression. The reasoning here is that if the function of the set, so the glutamate or the GABA pathway signaling, underlies the cortical thickness difference, the mean expression and cortical thickness difference correlation coefficient should be different for that of a random set of genes. And we test this by creating an empirical null distribution, where we take a random set of genes that is the same size as the gene set that we are testing, and we repeat this 10,000 times and take that as the null distribution that we test our gene set against. First, we created the cortical thickness difference profiles, and as mentioned before, our cortical thickness data was processed using FreeSurfer as well, so it was mapped onto the same atlas as the Allen Human Brain Atlas, giving us 34 brain regions per hemisphere. The gene expression data is only available from 2 donors in the right hemisphere, whereas the left hemisphere have data from all 6 donors. So for that reason, this analysis was only done using left hemisphere data. And since we're interested in understanding differences in autism, we calculated the cortical thickness differences by subtracting the average cortical thickness in each brain region of the non-autistic participants from the average cortical thickness of the autistic participants. And then we have our one value per brain region of cortical thickness difference across the whole data set. Then we created the gene expression profiles. And the first thing we do here is filter genes that have consistent gene expression so that we can be more confident that the gene expression we include is not biased. This is done in 2 steps as described in their 2018 paper. So when French and Paus created the DK atlas of the gene expression, they had expression data from each gene for each donor, but they also calculated the median. And with this, they provide the correlations of the expression for each donor to the median. And here we select the genes that have a donor to median correlation of rho is over 0.446. And this is a Spearman correlation coefficient. Then we use gene expression data from another atlas, the BrainSpan atlas, and this has a very wide age range of donors. So we select the ones with the same age range as our own cohort, so 6 to 30 years old, which gave us 9 donors. And here we calculate the donor to median expression values in the 11 cortical regions that also overlap with the ones available in the Adult Human Brain Atlas data. And we select the genes that correlate between the atlases higher than R is 0.52. This gave us 2,293 genes. In our glutamate set, we had 29, and we had 42 in the GABA gene set. And this is then what we use as our median expression profiles across brain regions that we then use as our interregional gene expression profiles in the analysis. Now it's time to start linking the gene expression with the cortical thickness differences, and we calculate the average correlation between the gene expression of the genes in a gene set with cortical thickness differences across brain regions. This gives us a distribution of correlation coefficients consisting of the genes in our gene sets, and that is the blue part in this figure. The average correlation coefficient across this is the dashed line in the figure. And we test this for significance against the empirical null distribution that we created, and that is the gray area in the figures. Then we do a two-tailed significance test to see if the gene set is significantly correlated compared to the null distribution to the cortical thickness differences. And this would then be indicated visually in this figure with the dashed line of the gene set average correlation laying outside of this null distribution. So if this dashed line would be within the gray area, that means that the gene set is not higher than chance level correlated with the cortical thickness differences. So now let's take a look at the results. So remember that I said that it would become relevant later that participants were between 6 and 30 years old. That is for now. So we know that cortical thickness changes a lot during development. So we divided our participants into groups of children, adolescents, and adults. And when we first ran this across the whole sample, there were no significant correlations, but we did see differences between the groups. So in children, there are still no significant associations, which you can see with the dashed lines laying within that gray box. But in adolescents and in adults, there were, but in opposite direction. In adolescents, this was positive, and in adults, this was negative, which you can also see in the figures where the dashed line in adolescents is laying on the right side and in adults on the left side of the null distributions. So this shows that increased gene expression of both glutamate and GABA is correlated with larger cortical thickness differences in autism, but how they relate is different across ages. And we can see the differences between adolescents and adults a bit more if we look at cortical thickness differences and gene expression in individual brain regions. So in the top row here, you see the differences of the cortical thickness differences in the 34 left hemisphere regions in adolescents and in adults. And in the middle and the lower rows, you see the gene expression of the most strongly correlated gene from each gene set. Red colors indicate a positive cortical thickness difference or gene expression, and blue colors indicate negative cortical thickness difference or gene expression. In adolescents, you see that the gene expression is quite similar to cortical thickness differences across brain regions, whereas if you look at adults, the gene expression is more in an opposite direction compared to the differences in cortical thickness. So again, this shows that increased expression of both glutamate and GABA genes are associated with larger differences in cortical thickness between autistic and non-autistic participants in adolescents and in adults. So glutamate and GABA functioning are affecting developmental differences of cortical thickness in autism. Taking all of this together then, what we have found is that glutamate and GABA play different roles linking to behavioral characteristics of autism, where glutamate genetic variation was associated to the ADOS and the ADI measures, whereas GABA genetic variation had a link to sensory processing. So there are different things going on here, showing that there are likely different glutamate and GABA balances affecting different autism characteristics. And we also saw that the expression of these genes linked to differences in brain structure, but also that these associations are different across development. And since autism is also in a way defined as certain differences in development, we see here that differences in development doesn't have to be just very early development or in childhood, but also that changes during adolescence into adulthood matters too. Now, this is not enough to know exactly what kind of imbalance is underlying what behaviors or exactly how this is mediated in the brain, but it's a good start to moving towards understanding these links better, and it gives us new questions for further research. I think it's important to note, though, that what we've done here doesn't tell us if there is more or less glutamate or GABA that links to brain structure or behaviors that we've seen here. Genes can encode for both loss and gain of functions that's involved in different processes. And here we didn't separate gene variants by function or anything like that. But what we can already take away from this is that it's relevant to take into consideration that balances between excitation and inhibition might affect behaviors differently, and that these effects differ across ages too. And given that there's been some inconsistent results from pharmacological studies in the last few years, this might actually be a factor that excitation and inhibition might be imbalanced in different ways, depending on what behavioral trait you're most interested in, but also that the age and development of the people you're working with affects these things as well. It might be that medications could be age sensitive, for example, but this is of course something that needs to be looked into in more detail. So moving forward now, we're currently collecting data in LEAP 3, where we get in vivo MRS measurements of glutamate and GABA concentrations as well. And with this, we can then look at genes and concentrations of glutamate and GABA at the same time. And there's also the preschool imaging study, or PIP, where we're collecting these same measurements, including MRS, in preschoolers, longitudinally as well. So then we can also look at even earlier development all the way into adulthood. And we can investigate how the relationships between these measures might change within individuals across development as well. And I think, and I hope you agree with me, this is really exciting and relevant things to continue to investigate so that we can get closer to actually be able to unpack what kinds of differences is actually underlying the behaviors and experiences of autistic people. And with that, I want to thank everyone that's been working on this with me, not just the ones that are named here, but everyone working on the cohort. And especially thank you to all of the participants, because without you, we cannot do this kind of work. And if you're interested in more details of this work, here is also a QR code to the paper on it. And thank you so much for your attention. If you want to reach me online, here's a bit of contact information, and I'm happy to answer any questions.